Hello, my name is Adrienne Lester, and this lesson today will be on ELISAs, also known as enzyme-linked immunosorbent assays. So the learning goal is that the learner or the student will be able to demonstrate how they can perform an ELISA assay and be able to interpret the results. Learning objectives for this lesson. Upon being given a protocol or instructions, the learners will be able to perform an indirect ELISA assay without much contamination and having 100% accuracy. The learner will also need to be able to describe the principle of an ELISA assay, differentiate between indirect and direct assay types of ELISAs, and to list the key components in their appropriate order that they are added to the reaction of the assay, and also to interpret the results upon completing the performance of the ELISA. Alright, and so we're going to watch this video.
The enzyme-linked immunosorbent assay, or ELISA, is an extremely sensitive test that is used to detect antibodies or specific antigens. The direct ELISA is a test for the presence of antigen. In this procedure, a known antibody is adsorbed to the inside of the well in a microtiter plate. After rinsing to remove excess antibody, the sample suspected of containing the antigen is added. Next, an enzyme-linked antibody that can react with the antigen is added. If antigen is present in the well, the enzyme-linked antibody binds to it and is retained. The colorless substrate for the enzyme is then added. Development of color indicates the presence of the antigen. In the indirect ELISA, antigen is added to the microtiter plate well, and the antigen attaches to the walls of the microtiter plate. After rinsing to remove excess antigen, the serum suspected of containing the antibodies is added. Enzyme-linked antibody capable of reacting with the constant region of other antibodies is then added, followed by addition of the colorless substrate. Development of color indicates the presence of the antibody being identified.
So now let's test your knowledge after watching that video. So question number 1, in an ELISA assay, what is produced in response to the substrate's conversion by the enzyme? A, color change, B, fluorescent compound, C, chemoluminescence, or D, optical reflection. And the correct answer is color change. Question number 2. An ELISA assay can detect the presence of A, antigens, B, antibodies, C, proteins, D, both antigens and antibodies, or E, all of the above. And the correct answer is all of the above. Question number 3 is a true or false question. In an indirect ELISA assay, does it involve, well, let me rephrase that. Indirect ELISA assays involve the enzyme-linked antibody attaching to a patient's antibody in their serum. And the correct answer is true. And question number 4 is a 2-part question. So what type of ELISA assay tests for the presence of an antibody? That would be an indirect ELISA, and one that test for the presence of an antigen is a direct antibody. ELISA, excuse me. So those questions sort of give a preview, well, a review on what the video says, but we're gonna go through the process of an indirect ELISA to see where those components play a role. So let's get started with that. So the first thing we're going to do is label the tubes for the appropriate serum samples that are going to be tested. Here in my simulation here, I have samples 1, 2, and 3, which are labeled 1, 2, and 3 respectively, and also I have positive and negative controls that will also be used. And we also need to label a microwell plate for all 3 of our samples, and also positive and negative control. As you can see here, we have each of the samples in triplicates in the plate, so that we'll have enough data for each of those. And so after we label, now we're going to add a known antigen to each of the wells. So we're going to add 50 microliters to each well. We're going to let that sit and incubate for 5 minutes. And after that incubation time, we'll take our microwell plate. We're going to dump out that antigen, any leftover antigen that's not bound to the plate at that point. And we're going to transfer buffer to each of the wells. And once they're filled, we're going to dump that well over onto paper towels and going to pat it until all the excess buffer is out of there. But when doing this, we want to make sure that we don't splash or spill any of the buffer from well to well, so we can cut down on cross-contamination. Contamination can cause there to be a positive result that's not really positive, so a false positive. And we want to do this wash step at this point 2 times to make sure that everything is washed away for our next step. So moving right along, so now to our microwell plate, we want to add 50 microliters of each of our samples, 1, 2, and 3, and our positive and negative control to its respective wells, which we labeled earlier. So as we do that, we're going to wait for 5 minutes and move on to our next step. So we're going to dump. So we're going to repeat that wash step like we did earlier. So we'll discard what we have in our microwell plate out on the paper towel. We'll transfer our buffer into the wells, and then we'll pat out the first set of buffer onto the paper towels, avoiding splashing or spilling over, and we'll do it a second time. Alright, and now we're going to add our secondary antibody, or I believe as the video said, the enzyme-linked antibody. So the secondary antibody, will be adding 50 microliters of that to each well, and the incubation time on that will also be 5 minutes. And after that incubation is complete, we'll wash wash the wells twice with the buffer solution. And last but not least, we add our enzyme substrate, and we'll be adding 50 microliters to each of the wells. The incubation time is 5 minutes, and from there it will be time to interpret and read and interpret our results. So to interpret the results, we have to observe a color change, and here is a simulation of what it would look like in the indirect ELISA assay. So we have our positive control that turned a blue color, which indicates if there is an antibody, the antibody of interest is present, that well will turn blue. As you can see, our negative control stayed clear, so that antibody isn't present, and so forth. So what does this mean? It means that sample 2 is positive for the antibody of interest, and 1 and 3 do not have that antibody in their samples. So now I want to test your knowledge on what we've discussed through this presentation. We have a few questions. So in this first question, we want to place the reactants of the indirect ELISA in the correct order. So first, we're going to add our known antigen so that it can absorb to the plate. Secondly, we're going to add our samples, which are in this case patient serum, to see if there's any antibody. The antibody of interest is included in their serum. Thirdly, we're going to add the enzyme linked antibody, or also called the secondary antibody. And last but not least, we're going to add our chemical substrate, and that substrate is going to, like I just previously said on the previous slide, that it'll cause a color change within the well, and that can indicate that the antibody is present. So post-test question number 2, well, what is the role of the chemical substrate when added? I already gave you that answer, but the correct answer is B. The chemical substrate will be converted by the enzyme to produce a color. In our example, the color was blue. Question number 3, what is one of the main causes of false positive results? Contamination, cross-contamination. This is a very sensitive assay. Any cross contamination by buffer being added too high into the wells and spilling over into the next one, or splashing while you're patting your plate on the paper towels can cause contamination, and it can possibly give you a false positive result, and you don't want that to happen. And this concludes the lesson. If you have any questions, here's my email address. Feel free to reach out to me if you have any questions or concerns.